Dear students, after studying this module, you shall be able to know about the chemical structure of pyridoxal phosphate, learn about various reactions carried out by different enzymes using pyridoxal phosphate as a coenzyme, understand pyridoxal phosphate as coenzyme in various reactions such as transamination, decarboxylation, non-oxidative deamination, transsulfuration and condensation, etc. Now let us introduce this topic of pyridoxal phosphate to you. The pyridoxal phosphate is a phosphate ester of aldehyde form of vitamin B6. Pyridoxal phosphate, or PLP as it is abbreviated, vitamin B6, which is also known as anti-dermatitis factor, includes various closely related forms of pyridoxamine, pyridoxal and pyridoxine. Here you can see the structures of pyridoxine, pyridoxal and pyridoxamine and their structure details. Now students, I would like you to pay attention on the active forms. Active forms are phosphate esters such as pyridoxal phosphate and pyridoxamine phosphate, or coenzyme, are formed by phosphorylation formed by enzyme pyridoxal kinase using adenosine triphosphate. Here you can see the phosphorylated forms of pyridoxal and pyridoxalamine. Now students, let us pay attention onto the pyridoxal phosphate. Pyridoxal phosphate is required by many enzymes catalyzing reactions of amino acid and amines. The reactions are numerous and pyridoxal phosphate is surely one of nature's most versatile catalysts. The story begins with biochemical transamination. In the year 1937, Alexander Bronstein and Maria Kritzmann in Moscow described the transamination reaction by which amino groups can be transferred from one carbon skeleton to another. The transamination reaction is a widespread process of importance in many aspects of nitrogen metabolism of organisms. For large number of transaminases, glutamate is one of the reactants. In 1944, Esmond Snell reported the non-enzymatic conversion of pyridoxal into pyridoxamine by heating with glutamate. He recognized that this was also transamination, and henceforth he proposed that pyridoxal might be a part of coenzyme needed for amino transesterases. The hypothesis was soon verified and this coenzyme was identified as pyridoxal 5-phosphate or pyridoxamine 5-phosphate. Let us now consider the non-enzymatic models for the same. Pyridoxal, or PLP as it is abbreviated, in complete absence of enzymes, not only goes slow on transamination with amino acids, but also catalyzes many other reactions of amino acids that are identical to those catalyzed by PLP dependent enzymes. Thus, the coenzyme itself can be regarded as the active site of enzymes and can be studied in non-enzymatic reactions. For such studies, Snell and associates drew the following conclusion, which basically says that the aldehyde group of PLP reacts readily and reversibly with amino acid to form Schiff bases, which reacts further to give the product. And here you can see the structure of pyridoxal phosphate in Schiff base linkage to enzyme lysine residue. Second conclusion was, for an aldehyde to be a catalyst, a strong electron attracting group, for example, the ring nitrogen of pyridine as in PLP, must be ortho or para to the aldehyde group. Now students, let us study about the variety of PLP dependent reactions. The reaction shown in general mechanism of action of PLP falls into 4 groups. Group A. These classes of reactions occur by loss of an alpha hydrogen. Dissociation of an alpha hydrogen from Schiff bases lead to a quinonoid carbanionic intermediate. The name reflects the characterization of two resonance forms. This intermediate ion can react in several ways such as racemization, transamination, beta elimination or replacement.
Some amino acid transformations at the alpha carbon that are facilitated by pyridoxal phosphate. As we have already told you earlier in this module, that pyridoxal phosphate is generally bonded to the enzyme through a Schiff base. Reactions generally begin with the formation of a new Schiff base, aldimine, between the alpha amino group of the amino acid and PLP, which substitutes for the enzyme PLP linkage. Now students, there are three alternative fates of this Schiff base: transamination, which is the fate A, racemization as the fate B, and fate C is decarboxylation. The Schiff base formed between PLP and amino acid is in conjugation with the pyridine ring. An electron sink that permits delocalization of an electron pair to avoid formation of an unstable carbon ion on the alpha carbon. The transamination route A is especially important in the pathways described in this particular module. The pathway represents only part of the overall reaction which is catalyzed by amino transferases. To complete the process, a second alpha keto acid replaces the one that is released, and this is converted to an amino acid in the reversal of reaction steps as shown in the figure. Pyridoxal phosphate is also involved in certain reactions at the beta and gamma carbons of some amino acids.
Now students, a proton can be added back to the original alpha position but without stereospecificity. A racemase which does this is important to the bacteria. They must synthesize D-alanine and D-glutamic acid from corresponding L isomers for use in the formation of their peptide glycan envelope. Transamination of amino acids. Amino transferases are classical examples of enzymes biomolecular ping-pong reactions in which first substrate reacts and product must leave the active site before the second substrate can actually bind with it. Thus, the incoming amino acid binds to the active site, donates its amino group to the pyridoxal phosphate and departs in the form of an alpha keto acid. The incoming alpha keto acid then binds, accepts the amino group from pyridoxamine phosphate and departs in the form of an amino acid. Now here you can see the sequence of the ping-pong reaction, or so to say a double displacement reaction, and how the pyridoxal phosphate is actually involved in it. Most amino acids undergo transamination reactions with few exceptions like lysine, threonine, etc. The mechanism of transamination showing the role of pyridoxal phosphate as a coenzyme is shown clearly to you here in this figure, and here the role of pyridoxal phosphate as a coenzyme is very crucial. Let us now study the third reaction, which is the elimination and beta replacement or the non-oxidative deamination. When a good leaving group is present in the beta position of an amino acid, it can be eliminated. A large number of such enzymes are serine and threonine dehydratases, which eliminate OH- and water. Here you can see the mechanism of such reactions. And the reaction mechanism of serine dehydratase is shown to you here clearly, with its role clearly depicted in the figure. Group B reactions, they include the removal of alpha carboxylate or decarboxylate. Several amino acids undergo decarboxylation requiring B6 as a coenzyme. Some important examples of decarboxylation are 5-hydroxytryptophan to serotonin, histidine to histamines, glutamate to GABA, and DOPA to dopamine. Now these are very important neurotransmitters or neurotransmitter based products which are responsible for the sensory perception in human body. The bond to the carboxyl group of amino acid substrate is broken in reaction catalyzed by amino acid decarboxylases. These also presumably lead to transient quinonoid carbanionic intermediates. Addition of proton at the original site of decarboxylation followed by the breakup of Schiff base completes this particular reaction sequence. Decarboxylation of amino acid is nearly irreversible and frequently it appears as a final step in the synthesis of amino acid compounds. Let us now begin the discussion of group C type of reactions which include side chain cleavage, that is the condensation reactions of amino acids. In this type of reactions, a reaction of side chain of Schiff base undergoes aldol cleavage. Conversely, students, a side chain can be added by beta condensation. The best known enzyme of this group is serine hydroxymethyl transferase, which converts serine to glycine and formaldehyde. The latter is not released in free form but is transferred by same enzyme specifically to tetrahydrofolic acid. And here is this mechanism, students, and the reaction. Threonine is cleaved to acetaldehyde by the same enzyme in this particular reaction which is shown to you here. Also shown to you here is the reaction mechanism of serine hydroxymethyl transferase and its function in the same. In the same group, ester condensation reactions join acyl group form COA derivatives to Schiff bases derived from glycine or serine. Succinyl coenzyme A is the acyl donor for biosynthesis of delta amine levulinic acid, an intermediate involved in the porphyrin synthesis. And here you can see the reaction which is actually showing you the conversion of succinyl COA into for the biosynthesis of levulinic acid. The enzyme does not catalyze decarboxylation of glycine in the absence of succinyl coenzyme A. And here you can see what is the role of succinyl coenzyme A and glycine in this particular reaction which is shown to you clearly in this figure. A similar reaction in biosynthesis of sphingosine serine is condensed with palmitoyl coenzyme A, an amino ketone intermediate. And here is the sequence for the conversion of the biosynthesis. Now let us begin our discussion with the group D conversions or group D reactions which involve the reaction of ketamine intermediates as electron acceptors. The fourth group of PLP dependent reactions are thought to depend on the formation of ketamine intermediates. In this form, the original alpha hydrogen of the amino acid has been removed and the C double bond NH plus bond of the ketamine is polarized in the direction that favors the electron withdrawal from the amino acid into the imine group. This permits another series of enzymatic reactions which are analogous to those of beta oxo acids. Both elimination and CC bond cleavage, alpha beta to the C double bond N group of the ketamine can occur. Now you can see this particular reaction happening in this figure very clearly from the quinonoid carbanionic form to the ketamine form and then leading to the pyridoxamine phosphate and alpha oxo acid. Enzymes of this group catalyze elimination of gamma substituent from amino acids. Eliminated groups may be replaced by other substituents either in the alpha or the beta positions. The ketamine formed initially by such an enzyme undergoes elimination of the gamma substituent, beta with respect to the C double bond N group along with the proton from the beta position of the original amino acid to form an unsaturated intermediate which can react in one of the three ways depending upon the enzyme. Addition of HY dash leads to gamma replacement, while addition of a proton at the alpha position leads to an alpha beta unsaturated Schiff base. The latter can react by addition of HY dash, or it can break down to form an alpha oxo acid, as I told you, and ammonium ion, just as in the beta elimination reactions discussed earlier on. An important gamma replacement reaction, students, is the conversion of O-acetyl, O-succinyl or O-phosphohomoserine to cystathionine. The cystathionine gamma synthetase reaction lies on the pathway of biosynthesis of methionine by bacteria, fungi and higher plants. Or subsequent reactions include beta elimination from cystathionine of homocysteine, which is then converted to methionine in this process, and this process is shown to you very clearly in this figure. Here you can see we begin with succinate and cystathione and including the pyruvate elimination, that is the beta elimination, forming homocysteine and then finally methionine. Now students, let us talk about glycogen phosphorylase and its role. While PLP is ideally designed to catalyze reactions of amino compounds, it was surprising to find out it as an essential factor for glycogen phosphorylase enzyme. The PLP is linked as a Schiff base in the same way as in other PLP dependent enzymes, but there is no obvious function for the coenzyme ring. The phosphate group probably acts as an acid-base catalyst. It has been estimated that 50% of the vitamin B6 in our body is present as PLP in muscle phosphorylase. Studies of vitamin B6 deficient rats suggest that PLP in phosphorylase serves as a reserve supply, much of which can be taken for other purposes during times of deficiency. Now have a look at the reaction and its mechanism here. Then we should shift our focus to pyridoxamine phosphate as a coenzyme. Let us discuss a little about this. If PLP is a cofactor designed to react with amino groups of substrate, might not pyridoxine phosphate act as a coenzyme for reactions of carbonyl compounds also? Have you ever thought about that? Now let us discuss about the same. An example of this kind of function has been found in the formation of 3,6-dideoxyhexoses needed for bacterial cell surface antigens. Glucose as cytidine diphosphate glucose, CDP glucose that is first converted to 4-oxo-6-deoxy-CDP glucose. The conversion of the latter to 3,6-dideoxy-CDP glucose requires PMP as well as NADH or NADPH. Now here you can see this mechanism for the conversion. Let us now consider the stereochemistry of PLP requiring enzymes. According to stereoelectronic principles, the bond in the substrate amino acid that is to be broken by a PLP dependent enzyme should lie in the plane perpendicular to the plane of the cofactor imine pi system. This would minimize the energy of the transition state by allowing maximum sigma pi overlap between the breaking bond and the ring imine system. It would provide the geometry closest to that of the planar quinonoid intermediate that is formed, thus minimizing molecular motion in the approach to the transition state. Another related reactions that goes through a ketamine intermediate is conversion of amino acid to kynurenine to alanine and anthranilic acid during tryptophan metabolism. Vitamin B6 as coenzyme in tryptophan metabolism is shown to you here along with its role. Now students, having discussed pyridoxine phosphate in maximum detail and its various aspects of synthesis and other uses, let us now summarize this module. Pyridoxal phosphate PLP is the active form of vitamin B6. PLP is formed from the phosphorylation of all three forms of vitamin B6. Vitamin B6 consists of a mixture of three different but closely related pyridine derivatives such as pyridoxine, pyridoxal and pyridoxamine. Pyridoxal phosphate acts as a coenzyme in a large number of reactions of amino acid metabolism. Some involve removal of one hydrogen or the alpha hydrogen. In this group, there are three types of reactions like transamination, racemization and deamination. Second group involves removal of one carboxylate as carbon dioxide, which involves all decarboxylases like DOPA, decarboxylate, ornithine decarboxylase, histidine decarboxylase and many others. Third group involves removal or replacement of sidechain or hydrogen by aldol cleavage, which involves serine hydroxymethyltransferase and threonine aldolases, etc. Fourth group involves reaction of ketamine intermediates involving cystathione gamma synthetase, anthranilic acid synthetase, etc. are other PLP dependent reactions. Pyridoxal phosphate is required for niacin coenzymes NAD+, NADP+ synthesis from tryptophan as told to you in this module. Also the enzyme glycogen phosphorylase contains covalently bound PLP.